So it's great to be here. Thank you, John. I think you said pharmacology, right? So I'm from the Department of Cardiology here in Göttingen. And what you see in the background of this slide, actually, is a human heart, which is not only red in the H&E state with cardiomyocytes, but it has all in between blue, the collagen, indicating cardiac fibrosis. And this has been my research area for the past 20 years. And actually, just a couple of years ago, I think my colleagues here at UMG realized that it's really important because their own patients, they realized there are patients with aortic stenosis who undergo TAVI and get aortic valve replacement. The main determinant of the outcome 1 to 2 years after TAVI is whether they have an amount of cardiac fibrosis over below the median when they have excellent survival or above the median. And this quantification was done in my lab. This is a patient with aortic stenosis. So finally, my colleagues agreed it's a really important thing, but there's currently no specific therapy and also no biomarkers. So what do these monozygous twins, Olivia and Isabella, have to do with that? Well, we know that in leukemia, the methylation, the promoter methylation of a single gene can cause it, even though these twins are otherwise identical on a DNA level. And this knowledge prompted us to hypothesize, or it prompted also my husband, who's a kidney fibrosis researcher, to hypothesize that this DNA methylation could play a role in fibroblast activation. And this is something we could show over 10 years ago, that indeed the methylation and silencing of fibrosis suppressor genes, just as there are tumor suppressor genes, can contribute to fibroblast activation and fibrogenesis. At that time, it was shown in the kidney. One of the genes we identified in that screen, it was an unbiased screen, genome-wide methylation screen, and one of the genes to be relevant was identified to be RASAL1. And what RASAL1 does, it shifts active RAS to inactive RAS, and if there is no RASAL1 because the gene is hypermethylated, obviously there is more active RAS and more fibroblast activation and fibrosis. Now, during my time as a postdoc and a junior faculty at Harvard Medical School in the Department of Matrix Biology, we would breathe that there's common mechanisms for fibrogenesis. And we would look at different organs. When one mechanism was identified in one, the other person who was in charge of kidney or tumor or lung or intestine would look for that. So obviously, when this RASAL1 was identified to be relevant as a methylated tumor suppressor gene, we not only looked in the kidney, but also in the heart. You see here a mouse model of ascending aortic constriction. And we saw that not only in the kidney, but also in the heart, it's hypermethylated. And the way we look for this is with methylated DNA precipitation. Methylated DNA is immunoprecipitated, and then we follow up with a PCR for the target gene. And we could show that this is not only relevant in the kidney, but also in the heart, first in the mouse model, but then we also looked in patient samples. These are patients with ICM or DCM, and in patients with high fibrotic hearts, there's a higher level of methylation of RASAL. So that was sort of borrowing knowledge from the kidney and seeing that it's also true in the heart. But we also had another case where we actually found a mechanism first in the heart, and that was the endothelial to mesenchymal transition, where we could show that endothelial cells under certain conditions can acquire a mesenchymal-like phenotype and contribute to fibrosis. And that was first shown in the heart and later taken to other organs, and it has been shown to be a global mechanism, essentially in any organ. Others have then shown, and we could show that this is blocked by a substance called bone morphogenic protein 7, and I will come to that later. Others then have shown that RAS not only increases fibroblast proliferation, but it seems to have a role in NMT, and we could show that also RASAL1 contributes to NMT. So with these two independent stories, we asked ourselves, on the one hand, we know that this fibrosis suppressor gene, like RASAL1, which methylation is induced by TGF-beta, it's relevant in the heart, in the kidney, and others have shown it's also relevant in the liver. And on the other hand, we could show that there is this bone morphogenic protein 7, which antagonizes TGF-beta in the heart and in the kidney. And we thought, well, maybe could this effect of BMP7 that we see, or that we had seen, be also a reversal of this methylation? And in order to look for reversal of methylation, I would like to just give you the background. Methylation is essentially a methyl group on the 5' cytosine, and you would think, well, you look for an enzyme that just removes that to be a naked cytosine, but there is no such enzyme in the human body, but instead the nature takes a detour here, and the detour is called hydroxymethylation. There's 3 enzymes, TET1 and TET2, TET3, which mediate this hydroxymethylation, and hydroxymethylated cytosine is in fact an inducer of transcription and eventually replaced by normal cytosine. And we asked the question, well, does BMP7 mediate this effect through one of the TET enzymes, and we identified TET3 as the enzyme that mediates BMP7 induced antifibrotic demethylative therapy. So one thing we asked at the time is, well, if TET3 demethylates the fibrosis suppressor genes, can we technically induce a gene-specific demethylation by bringing the catalytic domain of TET3 to a certain gene? And this work was done by Xingbo and Xiaoying in the lab. Xingbo is now an independent investigator, and what they did at the time, they mutated Cas9. It wasn't available at the time, the mutated version, in a way that it would not cleave anymore, but just guide, just bring the guide RNA to the target gene, and they fused the catalytic domain of a high fidelity, of TET3 to the gene of interest, and they showed it for several genes. Here I just show you for RASL1, they screened several guide RNAs here, 1 to 5. They saw 4 and 5 had an effect in fibrotic fibroblast cells to bring back the RASL1 expression. And when we looked at the methylation, indeed, for guide RNA4, methylation was reduced, and this was accompanied by an increase in hydroxymethylation. This was then also taken to the in vivo situation, all in a lentivirus, because the construct was so big. And so the lentivirus was tested again in a kidney mouse model because it's easier, it's faster, a 10-day mouse model where one of the ureters is ligated, and that leads to fibrosis basically after 5 days. And you see in this model, when we give this lentivirus, which has our RSG guide RNA with the Cas9 TET3 catalytic domain, we bring up the RASL expression, we reduce the methylation, we induce hydroxymethylation, and we also see a reduction in fibrosis. So we thought that's great, and we took quite some effort to also transfer the technology to an AAV, which is not so easy because it's a large construct. So we started using a split intein concept from bacteria, and it was quite successful. The reason I'm not showing it to you is because we also saw that there's more to TET3 other than just demethylating something. And this concept came from Sandip. He took up a finding we had in our lab which puzzled us, because we saw that in the mouse, or also in the human cells in cell culture, fibrotic fibroblasts show increased proliferation despite increased DNA damage, which is something odd. So you see here a mouse model of angiotensin 4, of angiotensin 2 after 4 weeks, and you see here the sham operated. We have a marker for DNA damage, which is gamma H2AX. Very few DNA damage in the sham operated, a lot of DNA damage in the hypertrophic fibrotic heart. And when we do a alpha smooth muscle actin staining with it, we see the increase in fibroblasts, and we also see that actually the DNA damage is in part located in the fibroblasts, and staining for proliferation, Ki67, also shows that there's an increased proliferation, and there has to be increased proliferation because obviously that's what drives fibrosis. And this is something that's not expected. Sandip found a paper which showed that TET3 actually regulates DNA damage response. And he postulated that maybe the DNA damage response is responsible for this proliferation despite DNA damage. And he used a reporter system. It was introduced right now by Christian Kupert for homologous repair, which is basically a non-homologous end-joining repair, and it shifts basically the fast and error-prone non-homologous end-joining back to the slow but error-free homologous repair. You see here in a TGF-beta treated cell system, this is, should be opposite. Actually, non-homologous end-joining goes up, and overexpression of TET3 rescues that. So, to put that together, we realized TET3 has more antifibrotic effects other than just the methylation or the reversal of methylation, but it can also shift the DNA damage response and thereby reduce fibroblast proliferation. So the question was, can we use that for therapy? And I told you, okay, methylation of our fibrosis suppressor gene induces fibrosis. If we bring TET3 to the system, like BMP7, we can reverse this, and Johnson & Johnson has tried to manufacture BMP7, but ultimately failed because it's difficult to make that. But there's another drug called hydralazine. You may know it in the US as aprisolin, and a very safe anti-hypertensive drug. And that also has TET3 inducing capacity and thereby demethylating capacity. And as a consequence, it has been used in several clinical trials as a cancer treatment, where one uses exactly that capacity. It has been shown by other groups that it inhibits DNA methylation transferases, and we have shown that it also induces TET3, the opposite enzyme, to make the effect even stronger. And when we looked in the literature, we actually found this old animal model in an isoproterenol-induced heart failure model, where they showed that SERCA2A also gets rescued with dehydralazine, and methylation of SERCA is reduced. Going back to the literature, we saw that in the 80s, there were actually studies in many thousands of patients where hydralazine was used in combination with isosorbide dinitrate, thank you, in comparison with placebo and with processing in thousands of patients with heart failure. And it had a significant advantage in survival as compared to placebo in processing. And as a consequence, but then later, in the next following years, the ACE inhibitors came on the market, and the ACE inhibitors were always just a little bit better as compared to hydralazine. So there was not really a financial interest in following up on the hydralazine. But there was a meta-analysis of this cohort, and one saw that it was especially good in blacks with heart failure. And that was then repeated over 10 years later, where again one saw that in African Americans with heart failure, these patients benefit from dehydralazine and this combination therapy. And as a consequence, it was the first race-based therapy on the market. Now you can ask why do African Americans benefit from it? And we hypothesized that it could be something about the metabolism of this drug, because the metabolism depends on whether you are a so-called fast acetylator or slow acetylator. You can divide the world in these two groups, and you see a study here which had nothing to do with medicine. They were looking into impact on hunters and gatherers on polymorphisms, and they made this study where you can see that up here in Europe, in general, we are statistically more slow acetylators, and down here, in general, you are more fast acetylators. So in theory, it could be that they metabolize it quicker and therefore have a lower dose. And indeed, we found that low-dose hydralazine is better in antifibrotic effect as compared to high dose. And that's in part because high doses of hydralazine induce HIF-1 alpha. So we hypothesized low dose, which is actually below the antihypertensive effect, is an optimal antifibrotic concept. and it also works in the mouse model of cardiac fibrosis with angiotensin II again. We see that low-dose hydroelacin reduces fibrosis. So we thought, okay, maybe our patients with high level of myocardial fibrosis at the time of TAVI could benefit from that. And we proposed to randomize patients based on their level of fibrosis by MRI. We proposed to enroll 300 patients to then be able to randomize to 150 patients and include them in, distribute them to randomize them to 3 arms, either standard of care or an established antifibrotic drug, spironolactone, or the combination therapy of spironolactone and dihydroelacin, and then follow up with a cardiac MRI 12 months later and see the outcome. And this proposal was actually successful. It was accepted as the 25th clinical trial by DZHK, who also fund this, or partially fund this symposium, and we made it to the local headlines. This is my colleague Miriam Puls, who is the attending and the clinician who actually does their aortic valve intervention, and together with me, she does this study. We received the funding decision last year. First patient was enrolled now in March 22, and yesterday we enrolled the 12th patient, and actually we randomized 6 patients, and we hope to have the results by 2025. Very few words on biomarkers, because I told you no specific therapy, but also no biomarkers. Again, we looked in the cancer field, and in the cancer field they detect circulating DNA fragments as a diagnostic guide, and we did the same thing. We did it with RASAL1 in a cohort that had no other comorbidities, no kidney disease, no fibrotic, no liver disease, and that was a cohort was made available to us from Mannheim with hypertrophic obstructive cardiomyopathy, who had an MRI-based myocardial fibrosis estimation, and we saw there's some correlation. We also had another cohort, HFpEF, followed up for 11 years, where we could judge mortality, and they're both circa and RASAL1 were associated with this mortality. Very briefly, we also look for new genes in an unbiased methylome-wide approach. We identified 4 new genes, and this is very preliminary data. Leon looked at the 100 patients, 100 TAVI patients where we had follow-up data, whether there's any correlation with survival, no correlation here, no correlation here, but for IRX3 and codin, he did find a correlation, and it appears that a higher level of methylation in the blood is associated with better survival after TAVI, even though we have no idea why. As a summary, I hope I could show you that aberrant DNA methylation contributes to fibroblast activation and fibrosis, that gene-specific demethylation via CRISPR-Cas9 is feasible, and that demethylating therapy to target cardiac fibrosis is currently evaluated in clinical trial, and obviously that circulating methylated DNA may be useful as biomarkers for progression of fibrotic disease. And with that, I thank you very much. This is an old picture. I used it because it has the Gänseliesel here to show you the tradition of Göttingen. Thank you.